Untangling PANDAS and PANS is a podcast about two little-known medical disorders characterized by the sudden and dramatic onset of symptoms such as obsessions and compulsions, vocal or motor tics, and restricted eating behaviors, and a whole host of other symptoms following a strep or other bacterial or viral infection. I have the privilege of interviewing some of the top researchers and clinicians in this rapidly growing area, known by various names such as immune-mediated neuropsychiatric disorders, infection-associated neuroimmune disorders, and autoimmune encephalitis, or simply PANDAS and PANS. My name is Dr. Susan Manfull. I am a social psychologist, the executive director of the Alex Manfull Fund, and the mother of Alex Manfull, who died at 26 years old due to PANDAS, a disorder my husband and I knew next to nothing about, certainly not that our daughter could die from it.
This is episode 8 of Untangling PANDAS and PANS, recorded November 23rd, 2024.
More than 2,000 years ago, the Greek physician and philosopher Hippocrates famously said, "All disease begins in the gut." Today, some researchers are taking a closer look at this proclamation. Today, we are thrilled to talk with Dr. Pawel Kiela, a leading expert in autoimmune and autoinflammatory disorders. We talk about his research on the gut-brain connection, particularly how the gut microbiome and microglia may play pivotal roles in the pathogenesis of neuroimmune psychiatric disorders such as PANDAS and PANS. All right, let's get started. We have a very special guest, Dr. Pawel Kiela. He has a doctorate degree in veterinary medicine, and he has a PhD in developmental physiology. The DVM degree is from the Warsaw University of Life Science in Poland, and the PhD is from the same university and Lund University in Sweden. He is now a PANDA endowed professor in autoimmune disease at the University of Arizona College of Medicine in the Department of Pediatrics. Drilling down a little further, he is in the Steele Children's Research Center. The Steele Children's Research Center is a very interesting place. It is an integrated program of basic research and clinical research, clinical care, and teaching to address a spectrum of neuropsychiatric disorders such as PANS, PANDAS, and Sydenham's Chorea. And I should mention before we go any further, I said that Dr. Kiela is a PANDA endowed professor. That is unrelated to PANDAS, as we're accustomed to using that acronym. PANDA in this case stands for People Acting Now Discover Answers. And it is a relatively small group of philanthropists who fund medical research to create healthier futures for children. So, Dr. Kiela, that's a long description of what it is that you do, but there's even more to what it is that you do. And I think that you're probably in a better position to talk about some of the centers of excellence in which you are a lead scientist. So can you elaborate on the centers of excellence under the Steele Children's Research Center?
Yes, of course. First of all, thank you, Susan, for having me on your podcast. I'm really honored to be a part of it. Yes, so within the Steele Children's Research Center, we have centers of excellence which are focused on addressing some of the specific disorders afflicting children. And I'm a member of two of them. I'm a lead scientist in the CPAE Center of Excellence, where CPAE stands for Children's Post-Infectious Autoimmune Encephalopathy, a term that we coined as an umbrella term that covers PANS and PANDAS. and related inflammatory and behavioral conditions in children. And I'm also a member of recently created Daniel Cracciolo Institute for Pediatric Autoimmune Disease Research within our center. So that it may seem like there may be some overlap between the two in terms of the autoimmune component, and that's where I thrive.
And you are also an associate director of a relatively new...
I'm the associate director of the Steele Children's Research Center for Basic Research and an associate director of the Pediatric Gastroenterology Fellowship Program, where I assist our fellows in developing and conducting research projects as part of the fulfillment of their fellowship curriculum.
Well, Dr. Kiela, I don't know when you find time to sleep. I
don't.
All right. Now, I know that your research, if I recall correctly, has a particular focus on Crohn's disease and ulcerative colitis, but there are many other subjects that you are interested in. Could you just give an overview of that now as well?
Yes. So my primary research is within the physiology and pathophysiology of the gut. And that includes studying chronic inflammatory disorders like Crohn's disease and ulcerative colitis, trying to understand the role of interaction between gut microbiota, bacteria that reside within our guts, with the epithelial cells that line and separate our organism from the countless bacteria within the lumen of our gut, and trying to understand how bacteria shape both innate and adaptive immune responses, not just within the gut, but also beyond. And for a long time, we've had a program also where we studied the link between gut inflammation and bone formation, and that also at some point led us to become really interested in the role of gut microbiota in shaping systemic immune responses and also neuroinflammatory responses in PANS and PANDAS.
So how did you make that leap to PANDAS and PANS?
So when we created the Center for Pediatric Autoimmune Encephalopathy, tasked with getting some preliminary data to see if microbiome of mucosal surfaces, and that was both nasal, throat, and gut, in some way correlated with the symptoms of PANDAS in our patients. And it was a sort of a leap of faith, simply based on an assumption that microbiome is a potent modulator of immune responses. And we wanted to see if there's a correlation, whether this can be exploited as a diagnostic tool or perhaps help explain some of the pathogenesis associated with neuroinflammation. And at that time, there was already quite significant amount of information published that indeed gut microbiota can play a role in shaping neuroinflammatory responses and neurodegenerative responses. And so it was not a completely insane idea, but we were operating largely in vacuum, thinking that could be the case.
What year about was that?
I think it was about 2018, 2019 when we started working on this.
Okay. All right. So I mentioned that the Steele Children's Center has several arms: the basic science, the clinical research, clinical care, and teaching. Was it in the clinic, were the clinicians already seeing patients that they were diagnosing as PANS and PANDAS, or is that what drove the question into your research? The clinical observations?
As you said, the mission of our department and the Steele Center is to combine clinical care with research. And that is one of the strengths of our department, which is the only pediatric hospital that has an associated research arm and research center. And we always try to, when we plan development of any clinic, we always try to merge and blend research, teaching, and clinical care together to continue, along that path of the combined strength of these three arms, and that every center that we create has a common mission, which is to heal, to teach, and to do research. So when the CPAE Center was created, we already knew that we want research to be a critical part of that center, and at that point, I joined that that center of excellence to see if we can do some meaningful translational research using mouse models and cells, and as well as human samples, to see if we can get some better understanding of the pathophysiology of the disease and maybe come up with better objective tools for diagnostic screening.
You really have the perfect background to move into that with your autoimmune and autoinflammatory research that had already taken place. You're in the pediatrics department, and you have a clinic there seeing a pediatric population. So that's a really fortuitous combination, I think, to have the research and the clinic right there. Lots of, as we said at our recent symposium, dialogue, I imagine, between the two groups.
Absolutely. I think we can deliver the best care by combining, efforts and close collaboration between clinicians and scientists. And that is really our long-term goal.
Okay, so I do have some questions about that, but let me just back up a moment and ask you what took you to Arizona? I don't know if you came from Poland or Sweden, but what took you to Arizona?
So after I graduated with my PhD, I took a postdoctoral fellowship position in the Department of Pediatrics, where I had an opportunity to continue in working in a much more mechanistic and molecular way on the developmental aspects of gut physiology. And that was in 1996. And I fell in love with Tucson, and I found the mentoring of my department head and my still father-like figure almost, Dr. Fayez Gishan, a huge asset, and the research environment at the University of Arizona was extremely helpful in growing, my career and moving along the ranks. And I think I was really, really lucky. The combination of great mentorship, access to resources, collegial and collaborative atmosphere here helped me really to get where I am today.
Well, and my husband and I had the pleasure of meeting you and touring the Steele Children's Research Center in December of 2018. And that was right after our daughter, Alex Manful, died. And as most listeners know, and I know you know, she died from complications having to do with her diagnosis of PANDAS. So there I met Dr. Daines, who is a pediatrician with special interests in allergy and immunology. Dr. Rice, Sydney Rice, who's a developmental pediatrician, and Fayez Gishan, your mentor, who is a pediatric gastroenterologist with a strong interest in nutrition. I hope I summarized their backgrounds accurately. And we met because you took my husband and me on a tour of your lab. We were very impressed with your large and very sophisticated lab. While we were touring it, you told me about some research that you were doing having to do with the microbiome in 3 pairs of twins. And we found that a captivating story almost in an area that we were just learning about. As I think you know, our daughter was diagnosed with PANDAS, but it was not long after she was formally diagnosed that she passed away. So we were hungry for more information, and we were fortunate to be able to visit your lab and the center in general shortly after she died. And we've been I remain grateful for that visit. So I'm wondering if we can begin by having you tell me about that study that you were doing at the time and that we, the Alex Manfield Fund, eventually contributed to.
Yes, so we started with an assumption that microbiome in patients with PANDAS may be different than from their healthy age and gender-matched controls, in some cases siblings. And we did that because microbiome or a collection of microbes that live on our mucosal surfaces and within the gut, as I mentioned, are very potent modulators of immune response in many different physiological and pathophysiological states. So we wanted to see whether nasal microbiome, throat microbiome, or gut microbiome changes in these children. And we found that it was pretty surprising to see that neither nasal and throat microbiome, which theoretically, these are the sites where the infection originates, were not that different. But gut microbiome, the entire microbial community of the gut, looking at fecal samples, was very different between healthy controls and PANDAS patients, and it could be especially visible among siblings that were discordant for PANDAS. So children that shared the same environment, household environment, presumably shared the same exposure to the same pathogen, shared the same diet, and yet their gut microbiome was very different. That was really eye-opening to us, and we realized that it may be very difficult to try to say that different gut microbiome is the precipitating factor in the disease onset, because this disease, as you know, has a very rapid onset, and catching patients prior to the onset to see if microbiome was already different is practically impossible. Especially that many of the PANS and PANDAS patients, as you well know, they are not properly diagnosed frequently for months from the initial onset of symptoms. So, but at the same time, we reason that changes in the gut microbiome, perhaps precipitated by eating disorder that often these children develop, could contribute to the establishment of chronic and relapsing nature of this disorder, and can still contribute to changes in immune reactivity and maybe also to neuroinflammation. And that was really the start of our work.
So your findings are particularly interesting because you were working with twins, in my mind. One identical set of twins and 2 fraternal.
Yes.
Just why would one develop a microbiome, as we're going to get into some of the differences, specific differences that you found, but why would the microbiomes be so different? You said diet, but they're in the same family. You mentioned eating disorders. Any other reasons why you'd see those the differences in the microbiome, but also the the stark difference in terms of one person having PANDAS and the other being quote unquote healthy.
Right. So to start with, each individual, even within the same household, they do have their own sort of fingerprint or, microbial signature within the gut. So we are very different in that respect, but probably not to the point that those differences would show as dramatic as we saw in these twins discordant for PANDAS. So I think that the reasons for that difference is pretty complicated, and I don't know if we can assign any one reason to that. Certainly changes in diet, and we know that these children do have changes in eating habits, very severe dietary restrictions, refuse to eat certain foods, and in somewhat similar to children with autism can be very, very picky in what they eat. So that diet being one of the strongest modulator of gut microbial composition most likely contributes to those changes. Another possibility is that a child who developed a more severe reaction to, let's say, streptococcal infection was given antibiotics and the other child was not. And the recovery from antibiotics may have been different because of a combination of eating disorder, maybe the length of antibiotic treatment, and so forth. So even though we made sure that our patients were not taking antibiotics for at least 4 weeks prior to sampling, we still saw those residual differences in microbial composition in the gut, which again is likely the result of a combination of factors, dietary changes, and perhaps poor recovery from antibiotics.
Interesting. And refresh my memory, please. At what point did you examine the microbiome? At what point into the disorder did you work with these patients? How long ago was the onset?
Within a year of diagnosis.
Okay. It really would be wonderful to do a longitudinal study.
Yes, and this is what we are doing right now with Juliette Madan at Dartmouth, looking at collection of stool samples in regular intervals to see whether we can capture maybe change in gut microbial composition at the time of, let's say, weaning from antibiotics and at the onset of relapse, because these children are probably, in analytical aspect, they serve as their ideal controls for themselves. It's just looking at the gut microbiome in remission and how it changes before the onset of a relapse.
Have you started that study?
Yes, we are actively collecting samples. It will take time, but we are beginning to accumulate samples from these patients.
And what are the ages of the patients? Is it just children, or are you including young adults, adolescents?
The oldest patient, and I would have to open up the file where we have this information. I think the oldest right now is about 14 years.
Okay.
Of course, this may change as we recruit more patients.
That's great. All right. Well, backing up to your original study there, I'm wondering if you have any ideas that you could share with us about why you didn't see differences in the nasal and throat samples and you did in the fecal samples.
I think it's actually a more difficult question to answer than when you actually do see differences. I can't really explain it why we don't see the differences.
Well, do you think the fecal samples are a more sensitive measure of what's going on? in the
I think
microbiome?
fecal samples reflect more because diet perhaps does not affect the throat and nasal composition as significantly as it does to the gut microbiome composition.
Okay. In the study with Dr. Madan, are you collecting are you collecting just fecal samples, or are you also collecting nasal and throat samples? Just out of curiosity.
I think we are focused on fecal samples. Here in Tucson, we continue to collect both throat and nasal swabs.
And will that research have any implications for using what do we call it? Fecal matter?
Transplant, fecal microbiome transplant. Potentially, I mean, it's a technique that is gaining traction right now. We have, I believe, 2 FDA-approved products to be used for recurrent Clostridium difficile infection, which is a often hospital-acquired infection, and fecal microbiome transplant is the best treatment for that. We have some preclinical and clinical trial data from patients with autism showing that fecal microbiome transplant can, to a certain degree, improve the symptoms. We also know that in autism, fecal microbiome and changes in fecal in gut microbial metabolic activity contributes to microbial metabolites that can promote aggression and anxiety in children with autism. So translating some of these findings to PANS and PANDAS may at some point lead to a clinical trial with fecal microbiome transplant, especially that we see that in mouse models, that when we transplant microbiome from PANDAS patients to mice, we definitely see higher inflammatory response, systemic inflammatory response in these mice. And we're also currently looking at how fecal microbiome from PANDAS patients, as compared to healthy controls, affects the brain function and expression of genes associated with inflammatory response in different regions of the brain. So hopefully we'll have some of the results to share with you in the next couple of months. If indeed we have convincing evidence that changes in the gut microbiome in patients with PANDAS result in a more pro-inflammatory type of microbiome in the gut, approaches that would hope to fix it or replace it with a more healthy community could potentially contribute to much better outcomes in treatment. Of course, fecal microbiome transplant is not an easy thing to do, and it can be a dangerous thing to do. It's not just the cost, but it's the fact that it's very difficult for scientists and clinicians in the field of gut microbiome. It's very difficult to define a healthy microbiome and what it is.
Oh, that's interesting.
So the source of the fecal microbiome transplant is extremely important, and making sure that we're not transplanting inadvertently something else. And I'm not necessarily talking about pathogens, although that is also a critical element of choosing what we transplant. But also as a community, gut microbiota can do a lot of different things, sometimes unanticipated things. So case reports have shown that you can transplant, let's say, acne with microbiome. You can transplant depression or susceptibility to depression with microbiome. So again, the definition of what a healthy microbiome is difficult, and identifying a microbiome that would be curative for any given patient is also quite challenging. But the field is getting there, and hopefully, within my lifetime, this will become a standard approach to a lot of different disorders.
That's really fascinating. I know our daughter was very interested in fecal transplant treatments, but didn't live long enough to explore them. When you mentioned a few minutes ago about transplanting in mice, I think you said you were transplanting microbiome.
Yes, so we take bacteria from the twins discordant for the disease,
Okay.
and transplant them into germ-free mice, so mice that are devoid of any bacteria or viruses or fungi within their body anywhere. So they serve as this empty reservoir that we can repopulate with bacteria from either other mice or from patients.
Okay.
And then we can study the outcomes of that.
And you found, I think you said, correct me if I'm wrong, that you found an increased inflammatory response.
Yes.
In those mice. And what was that measured by? What, as evidenced by?
So we looked at production of cytokines by immune cells locally in the gut and in mesenteric lymph nodes, but also systemically in the spleen.
I guess my next question is, what were some of the main findings in the twin study? And one of them, I believe, had to do with the increase in cytokines when the microbiome was transferred. Or am I mistaken and I'm referring to the mice study?
No, you're right. I mean, the main findings was that twins discordant for PANDAS had very different microbial composition in the gut, and the transplant of bacteria from these discordant twins to mice resulted in very different immune response, where mice transplanted with microbiome from PANDAS had more aggressive immune response.
So I have here in my notes related to 20 or more cytokines that were increased if the microbiome was transferred.
Correct.
And IL-17 was yeah. So there's a lot of research right now on IL-17.
That's correct, because it's one of the pro-inflammatory cytokines that is capable of disrupting blood-brain barrier that allows the influx of autoreactive antibodies to the brain parenchyma, but potentially other compounds from circulation, including perhaps compounds of microbial origin that can contribute or promote neuroinflammatory responses. And this is also something that we actively investigated, because we did untargeted metabolomics of both fecal samples and serum samples from patients with PANDAS and healthy controls. And it's a method that allows us to look at several thousands of different metabolites, both of microbial origin as well as of human origin, dietary origin, and look which ones are different. And we found pretty substantial differences in metabolic composition, both in the fecal samples, but perhaps even more so in serum of patients with PANDAS. And this was fascinating, because about half of those metabolites that were differentially abundant in patients with PANDAS can also be found in a database of microbial metabolites. That doesn't necessarily mean that every one of those compounds is exclusively produced by bacteria, because there's a considerable overlap. Some metabolites are produced by the host, or humans in this case, and by bacteria. But the fact that such a large proportion of differentially abundant metabolites could be potentially attributed to changes in the gut microbial composition and metabolic activity may actually be very relevant to the pathogenesis of the disease, especially in the context when blood barrier function is compromised, and potentially many of these compounds, which normally would not be able to penetrate into the brain parenchyma, now they can.
Okay.
that's what prompted us really to look at what happens if we expose human microglia to sera from patients with PANDAS or healthy controls. Would we be able to see activation of these microglia, which could contribute to neuroinflammation?
Okay, put a pin in that thought for a moment. I've got, I have a couple more questions on this, and so just to put this in a larger perspective, the IL-17 is a cytokine that's investigated in other areas beyond PANDAS and PANS. One is Crohn's disease, if I recall correctly, which is that right, or am I mixing up something?
So interleukin-17 is a very important mediator of inflammation in IBD, including Crohn's disease.
Which you studied initially.
Correct. Yes. Although clinically, it's not the most useful target, because interleukin-17 is one of those, as there's a lot of them in biology, double-edged swords.
Yes.
Where,
can you elaborate?
Small amount in the right place is protective. Too much of it for too long and in the wrong place is detrimental. And targeting IL-17 in inflammatory bowel disease turned out to be, in some cases, detrimental and at best not clinically viable, or it didn't have any positive outcomes.
In PANDAS, it is an educated guess that IL-17 modulators might be useful in a subset of these patients, correct?
Yes, yes, we talked about it at our last meeting in Portsmouth. Yes, there's certainly a lot of interest from clinicians in targeting IL-17 for the treatment of PANS and PANDAS. And it's definitely an area worth exploring, especially that there are products on the market that can be used off-label to test it in clinical trials.
Yeah, so it's a really exciting time. All right, and one more question with regard to your twin study. It in your you presented the study at our first symposium, and there were a lot of oohs and ahs in the audience when you identified some of the bacteria, some of the differences in the number of bacteria that were present in PANDAS versus not PANDAS patients. Can you elaborate on that a little bit?
Yes.
Maybe even backing up, like the normal microbiome is, the normal healthy one is very diverse. Correct.
So loss of diversity is a very common feature of any chronic inflammatory disorders, and we know that it's part of the problem. I often compare the diversity of microbiome to diversity of your retirement portfolio. That the more diverse your portfolio, if the market takes a hit, your portfolio will be less affected or will recover easier. Whereas if you invest in 2 or 3 different stocks and the market goes down, you could be very severely affected and have a difficult time recovering your retirement fund. It's quite similar with gut microbiome. Is the more diverse the ecology of the gut, the more resilient it is in the face of an environmental insult, be it an infection, be it antibiotic treatment, or anything.
However, we could fix our stock portfolio by purchasing stocks in different areas. It doesn't translate quite as well in yet, it seems to me, in buying supplements for the microbiome. Or am I wrong?
No, you're right. I mean, it is a very active area of investigation. Unfortunately, it's somewhat polluted by companies that try to market to people directly, often without good scientific support for some of the products. The field of probiotics, prebiotics is really quickly evolving, and we're beginning to understand that not only the traditional probiotics, which are usually like milk fermentation products associated, like the bacilli and other bacteria, do not really do as much good to human health as the manufacturers would want us to think. But we also realize that prebiotics, which are often soluble and insoluble fibers that we are also bombarded in the ads about their health benefits, they may actually be, their effectiveness may be different depending on an individual. And so I guess what I'm trying to say is that there's no one solution that fits everybody, and it is a lot more complex than we all wish it was. But certainly there's a lot of research also in developing new generation probiotics, which are bacteria derived from the gut of healthy patients, well characterized for their beneficial effects in preclinical and clinical studies, and using them in combination. So these would be bacteria that are more capable of colonizing the human gut and staying there for an extended period of time, if not permanently. And of course, we have the emerging field of fecal microbiome transplant that can help to increase the diversity of gut bacteria.
So one of your findings was that, I'm going to butcher this word, I'm sure, but Akkermansia, a bacteria by the name of Akkermansia, was distinctly Akkermansi. How do you pronounce that?
Akkermansia mucinifila.
Okay. So there was a significant difference, a significant depletion, or maybe we don't know if it was depletion, but a significantly lower amount of this bacteria found in the gut microbiome of those with PANDAS. And so our audience in that first symposium asked you, can we go out and buy that bacteria? Can you tell us why that's not going to work now?
I'm not saying that it won't work. We just don't have enough data to support this. And it's also a bit of a risky adventure to do it, even though you can go on Amazon and purchase Akkermansia mucinifila probiotic supplement. And the reason I'm saying is, yes, we see a decline in the abundance of Akkermansia mucinifila species and strains, which is especially dramatic when we compare twins discordant for the disorder. The reason why I say that it may be a little dangerous is that the scientific literature on the role of Akkermansia mucinifila is still evolving, and there are contrary information about benefits versus risks of Akkermansia supplementation and their role in the disease process. One of the studies that was somewhat related to PANS and PANDAS was a study with patients with multiple sclerosis that showed that these patients actually have higher levels of this bacteria in their guts. And the initial conclusion of the authors was that since it's higher, it probably contributes to the disease. But then a follow-up paper from another group showed that it actually may be a response that is aimed at counteracting the disease process, trying to actually reduce inflammatory response, aimed at the central nervous system, because in a mouse model of multiple sclerosis, they showed that supplementation with Akkermansia was able to at least partially attenuate the disease. Now, follow-up studies often related to intestinal inflammation show that you can see both beneficial and detrimental effects of Akkermansia, which may be dependent on the strain of this one bacterium that you're supplementing, where some of them show beneficial effects and some detrimental. And since there's more than one strain of Akkermansia muciniphila in the gut, it may be really important to know which one we are trying to supplement. The other issue with over-the-counter supplementation with Akkermansia is that this bacterium is an obligate anaerobe. What it means is that it lives only in strictly anaerobic conditions, without any exposure to oxygen, and even very short exposure to a small amount of oxygen kills them. And because it's not a bacterium that creates spores that would allow them to survive normal environment in which we live, I would love to see some evidence that the probiotic that is being produced by these companies and sold over the counter contains actually viable bacteria. It's hard to imagine for me that during the manufacturing process, these bacteria are not exposed to oxygen, and if they do, they're most likely dead. Having said that, I cannot possibly exclude a possibility that even dead bacteria could serve some sort of protective immunomodulatory effect in the gut, but there's really no good clinical or preclinical data to show that yet.
And this just underscores again how complicated these disorders are, and nothing can be fully understood in a vacuum. It has to be seen in context.
Yeah, absolutely.
All right. Well, still just wrapping up that very important study. Your preliminary data, in that study, the thought emerged, at least, maybe you can elaborate on this part, that the PANDA's systemic microbiome is associated with the activation of microglia. So that is our transition into your recent research that you presented at our symposium. Can you talk a little bit about what led you to think that, and what exactly microglia are?
Right. So microglia are what immunologists consider to be brain-resident macrophages. So they're part of the innate immune system, innate meaning that they don't develop memory. They're part of the ancient conserved immune response mechanism. But these cells have evolved really to play a lot of functions in the central nervous system, where they, under normal circumstances, they contribute to synaptic trimming, to healing from any type of injury, cleaning up debris. So they do have a lot of positive function, but when exposed to stimuli that normally they are not exposed to, they can become pathogenic, and they can promote tissue damage, inflammatory response, and chronic neuroinflammation. And activation of microglia is a very common feature of most, if not all, neuroinflammatory and neurodegenerative diseases. And why we became interested in these cells is it's because of the metabolomic analysis that I mentioned earlier that we did in the serum of PANDAS patients, which identified a lot of compounds changing in the circulation that could be of microbial origin, could be of host origin, but compounds that potentially, in the context of impaired blood-brain barrier, can actually penetrate into the brain parenchyma and activate microglia to push them out of their normal homeostatic, if you will, or healthy state into a pathogenic state of activity. And so we wanted to work with human microglia to make this more relevant to human disease, and that's why collaboration with Eric Blackman from Albert Einstein College of Medicine was so helpful, because he's a true expert in modeling human disease using pluripotent stem cells which can be forced to differentiate into different specific types of cells, including microglia. So we learned how to use these inducible pluripotent stem cells and push them to differentiate into hematopoietic stem cells initially, and then to microglia. And then we expose these cells to sera from patients, from PANDAS or healthy controls, and we looked at their activation status.
And what did you find?
It was really striking that that serum from patients with PANDAS was consistently much more prone into pushing microglia towards inflammatory state. And what we also found was that some of the changes that we observed, particularly expression of the typical markers of activation of microglia, which are associated with inflammatory state, was dependent on endotoxin or the receptor for endotoxin, toll-like receptor 4. In that if we scavenge endotoxin from serum, in this case we use an antibiotic that can bind to endotoxin and prevent it from binding to TLR4, it's polymyxin B, or using antibodies that bind to the receptor for this endotoxin, or toll-like receptor 4, TLR4, we were able to attenuate this response and expression of activation markers on microglia. And to us, it also added an additional line of evidence, or at least a strong suggestion, that patients with PANDAS, in addition to changes in the gut microbiome, may also have a defect in gut permeability, or a leaky gut, if you will, that allows endotoxin, which comes largely from gram-negative bacteria in the gut, to translocate to the circulation and then activate downstream inflammatory signaling pathways. Importantly, endotoxin is also a very potent substance that breaks down the integrity of the blood-brain barrier. So perhaps a combination of inflammatory mediators such as interleukin-17 and the presence of endotoxin in the blood contribute to the breakdown of the blood-brain barrier, and then endotoxin can continue to activate microglia towards more pro-inflammatory state, and then promote neuroinflammation and all the behavioral changes that we see in those children. So I think it's going to be, in the near future, is going to be really important to create this integrative approach to PANS and PANDAS, where, scientists and clinicians can work together on identifying the entire pathway. That we don't work in silos, but rather collaborate and try to create this integrated picture of what happens holistically in the body of patients with PANDAS. Working on the autoimmune aspect and finding autoantibodies towards basal ganglia is not done in a vacuum. That it's done in the context of damaged blood-brain barrier, the role of microbial compounds, and the role of gut microbiome in altering intestinal barrier function that allows these compounds to cross the otherwise impenetrable intestinal barrier. And then the next stage, for these compounds to cross the otherwise impenetrable blood-brain barrier, and then lead to downstream inflammatory responses in the brain parenchyma. I think it's going to be really, really important that we all work together and we try to holistically approach all of those findings.
That's fascinating. Can we just drill down a little bit for those of us who don't use this language all the time? Can you more or less explain what an endotoxin is? Can you provide a definition now for it for us?
Yes. So endotoxin is a lipopolysaccharide that is a compound that is part of a bacterial wall. A majority of endotoxin-producing bacteria are in the gut. They reside there permanently. But endotoxin does not get across the intestinal barrier unless it is disrupted by some insult.
Like
intestinal inflammation.
Okay.
So,
all right.
And so if changes in gut microbiota in these patients lead to even a low-grade inflammation in the gut and increased permeability or leakiness, some of these microbial products that are normally sequestered to the lumen of the gut and expelled with feces, with poop, may get across into the circulation. And lipopolysaccharide may be one of them.
And what is that?
That's endotoxin.
Oh, that's the endotoxin,
that's
right?
the endotoxin.
Okay.
And,
all right.
So lipopolysaccharide works on a lot of immune cells, but also on epithelial cells. It practically affects the function of every cell in the body that expresses a receptor for endotoxin. And that receptor is called toll-like receptor 4, or TLR4. And that's what I was referring to, that if we block the function of this receptor, to block the ability of endotoxin to bind to it, we can partially restore the expression of activation markers on microglia. But it's not all of the story, because we see a lot of other changes in microglia, pro-inflammatory changes that are not dependent on endotoxin. So there's clearly multiple signals within the soluble fraction of proteins or other type of metabolites in circulation of PANDAS patients that can affect microglia and push them into this more pro-inflammatory state. And one of our goals is to identify what they are.
What the other factors are
Correct.
that push microglia into the inflammatory state. All right. So it's frequently observed in children and adolescents and young adults who have, I'll stick with just saying PANDAS, who have PANDAS, that if they become sick, a few days later, they may have a flare. Could this be related to the endotoxin, a bacteria that gets into the system, and the endotoxin in the gut, or am I just being a real layperson here? Is there any kind of connection with that observation that is pretty common?
At this point, I don't know if we can point to endotoxin as the only one, component of the relapse. Like I said, that it's probably much more complex than that. Although, in a way, your question is correct in that our hypothesis is that changes in the gut microbiota that children with PANDAS develop as a consequence of the disease or perhaps treatment contribute to establishment of the chronic and relapsing nature of the disorder. And again, the gut microbiome and intestinal barrier function may not be the initiating factor, but they may be the sort of the second hit that helps establish the chronicity and relapsing nature of PANS and PANDAS. And maybe we can intercept that, and by targeting gut microbiota and gut barrier function to prevent relapses, because we don't know how to prevent the disease, right?
Mm-hmm.
In terms of its acute onset, and we don't really know at this point what makes one individual more likely to develop it and not, and the other individuals do not. We don't know the answer to that. But once the disease is diagnosed, can we come up with ideas of how to maintain remission and prevent relapse? And maybe in that context, the role of the gut and gut-brain axis in PANDAS may be a very attractive target.
So I'm not sure how to articulate this question, but how, knowing that there is can be acute changes as the disease ebbs and flows, and the individual can have changes in behavior and neuropsychiatric symptoms very quickly changing. The endotoxin and the TRL4 contribution in getting out of the lumen, is that correct?
Yeah.
And then causing the blood-brain barrier to be more permeable. Is that something that happens quickly?
It can. Now, I don't know in the context of PANS and PANDAS if that happens quickly, but in the context of intestinal injury and inflammation, it can happen very quickly. Now, because we know that usually PANS and PANDAS are precipitated by a respiratory infection. I'm not sure to what extent acute changes in the gut function can contribute, to the original symptoms. It's not impossible that they do, but again, because of the nature of the onset of this disease, it could be difficult to study. Certainly if we had access to patients who just presented with the disorder, patients that were not, that did not go to multiple doctors, that ignored the symptoms or decided to treat it as a psychiatric condition, but rather, patients that can be immediately diagnosed, that we can also study their intestinal permeability, gut microbiota at this point, and so forth. I think we would gain a much better knowledge of the early events associated with the disease development. And hopefully, as the medical field becomes more cognizant of the problem, we may be able to go earlier and find out more about the early events associated with this symptom development.
I wonder if a worthy substitute to what you've just said, which, as you've also said, would be difficult to see that patient very quickly after the onset of the initial symptoms. I wonder if in the patients that are being seen, if they're exposed to strep, for example, if they come in and have their microbiome, for lack of better words, analyzed at that time to see how it changes. And it would be particularly interesting if that did precipitate a flare, like what happened in the microbiome.
Right. So the best substitute for early detection and early sampling is longitudinal sampling in patients with chronic PANS and PANDAS.
Right.
And I think we talked about it earlier that can help us to establish a baseline in microbiome and gut permeability during remission, and perhaps capture the events leading up to a flare.
For example, if they've been sick.
Right.
Which, yeah. Just for the record, Alex was sick 5 days before she had a flare. And I would venture to say that she had never had anything remotely similar happen to her. She did not have flares in such an extreme case, but she was sick 5 days before that. This is all incredibly interesting. I just thought I would share with you, as you know, we donated Alex's brain, and it eventually is resting at Georgetown University Medical Center in the POND Brain Bank. POND stands for PANDAS, PANS, and other neuroimmune disorders. And her brain had 2 autopsies, or this final anatomic diagnoses. And her brain was diagnosed, they observed mild gliosis of the caudate nucleus and the thalamus. And the gliosis that was found in the caudate nucleus is, again, I don't know how to pronounce the word. It's parenchymal.
Mm-hmm.
Parenchymal astrogliosis of the caudate nucleus. And reading that, I'm wondering if some of your research in the future might involve brain tissue.
So we are certainly looking at the brain tissue in mice, as I mentioned. We want to know how bacterial community from PANDAS patients contributes to changes in the expression of genes in the different regions of the brain, and we plan to. We're doing it right now, actually, to use a technique called spatial transcriptomics, which allows us to look at large gene expression panels and looking at gene expressions at the level of single cell within the known architecture of the brain, so that we can define which types of cells and in which regions of the brain develop changes in gene expression that we can then perhaps attribute to an ongoing inflammatory process.
Mm-hmm.
So if this is successful, then perhaps working with human brain from Georgetown repository would be the next step, where we could utilize perhaps even a section of Alex's brain, particularly from the region of basal ganglia, and use spatial transcriptomics and compare that to gene expression profile in the brain of maybe a victim of accident. I don't think Georgetown has brains from sort of healthy controls.
They do. They do.
They do. Okay.
Mm-hmm.
So that would be very helpful, and we can get a glimpse not just at morphological differences between human brain with PANDAS, but also molecular differences that could help us maybe define really what's going on there at a molecular and single cell level. It's still a very costly technique. I believe that the work that we are doing right now in mice will cost us upwards of $20,000, and I would imagine that comparing human brains would be quite similar to that. But it may be a very important experiment to do, and could yield potentially a lot of useful information.
So she had perivascular astrogliosis in the thalamus. And I know this is astrogliosis as opposed to microgliosis. And there's what, 5 different types of glial cells, something like that.
Yeah.
What are the astroglia cells informative with regard to, or as informative as microglia with regard to neuroinflammatory disorders?
Absolutely. Astrocytes are also critical components in inflammatory response. But what you're describing are histological findings. And histological findings are very important for diagnostic purposes, but you miss functional changes that may be equally important as increasing numbers of astrocytes in a particular location. So microglia may be in an activated state, and yet you won't really see that on an H&E or hematoxylin and eosin stained slide, which is the typical way that pathologists look at tissue sections.
Okay. Well, I can talk to you later about that, but I was just curious. So the microglia has certainly, or the views of microglia have certainly changed in recent years. You and I talked about this book, The Angel and the Assassin, by Donna Jackson Nagizawa, and which is basically about the history of microglia use in science, I think we could say, and particularly relying on Beth Stevens, who is a neuroscientist in the Boston area. And I'll just to put it in perspective for how much things have changed for our listener, I'll read you an excerpt from the book, which says, until recently, microglia were thought to be merely the brain's housekeepers. Later in the book, it refers to the microglia as the garbage men or something like that. It helpfully removing damaged cells. But a recent groundbreaking discovery revealed them to be capable of terrifying Jekyll and Hyde behavior. When triggered, and anything that stirs up the immune system in the body can activate microglia, they can morph into destroyers, sparking a wide range of problems from memory loss and anxiety to depression and Alzheimer's, and in our conversation, possibly PANDAS and PANS. Under the right circumstances, however, microglia can be coaxed back into being angelic healers, able to repair the brain in ways that can help alleviate symptoms and hold the promise to prevent disease. Does that make sense? Does that resonate with you?
That is a wonderful quote, and it really encapsulates not just the changes in the scientific view of microglia, but changes in our view of the brain as a whole, which we used to think of as an immune privileged organ, where it's always under every circumstance protected from the outside influence of inflammation and immune cells and antibodies and all of that. And the view of the brain and the immunity or immunology of the brain has really gone a long way since those textbook assumptions. More and more frequently we realize that perhaps many, if not most, of the psychiatric disorders, or what we considered psychiatric disorders, have neuroinflammation as the, as a fundamental base or cause of symptoms. How many of them are induced by infection, viral, bacterial? We don't know, but I would imagine that PANS and PANDAS are just part of a large sort of syndromic response where the brain is attacked as a bystander in response to infection. We see more and more cases in clinics, and I'm sure when you speak with Juliet, she will speak to that as well in patients with COVID. That also are suggestive of post-viral neuroinflammatory responses. It's an evolving field, and it's a fascinating field to follow. And I'm a little scared as a GI scientist to venture into this area, but I hope I can find ways to contribute something meaningful to it.
Well, I'd venture to say you already have. And I think that that's a great way to conclude our talk. I don't think it's an exaggeration on my part to say that our conversation has been really riveting to me and cast a lot of light on some of the things that, or some of the connections that I was unable to make without this conversation. So, Dr. Kiela, I thank you very much for talking with me today.
Thank you, Susan. It's been a privilege and honor, and I appreciate the organizing of your symposiums, which has helped our group tremendously in creating networks of collaborators and helping each other in the field, but also for your initial support and the seed grant that helped us look at some of the autoantibodies in PANDAS, and you've been a champion, and it's really a privilege to be working with you.
Thank you. I would say the privilege is mine, and again, we're just very grateful for what you're doing. So thank you.
Now it's time to start publishing our work.
This is true. All right. Thanks so much, Pawel.
Thank you, Susan.
This concludes episode 8 of Untangling PANDAS and PANS. Thank you for listening. For more information about PANDAS and PANS and the Alex Manful Fund, please visit thealexmanfulfund.org. The content in this podcast is not a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified healthcare provider with any questions you may have regarding a medical condition.